Welcome back to the deep dive. Today, we are undertaking really a critical deep dive into a topic that sits right at the center of emergency medicine, transplantation, cardiac care, ischemia-reperfusion injury, or IRI.
That's right. And it's a subject defined by this profound, almost frustrating biological paradox.
We're discussing the mechanisms behind the damage, the actual cell damage sustained during life-threatening events like a heart attack, myocardial infarction, or a cerebral stroke. You know, when tissue is starved of blood and oxygen, the damage starts, well, immediately.
Yes, and the stakes could not be higher. We're talking about the core molecular processes, the absolute nuts and bolts that determine whether millions of neurons in the brain or cardiomyocytes in the heart survive or die.
But here's the central paradox, the one that keeps clinical researchers up at night. We have to restore blood flow to save the tissue. There's no question about that.
Absolutely necessary.
Yet that sudden, necessary restoration, the rush of blood back in, can paradoxically trigger this destructive cascade. It can kill cells that were previously only reversibly injured, cells that might have made it.
It's the ultimate irony, isn't it? You successfully remove the blockage, the oxygenated blood rushes back in. That's the moment of clinical triumph, right?
You'd think so.
But it's precisely then that a secondary internal chemical war begins. This additional loss of cells, that's the reperfusion injury. It is, in many ways, the final hurdle to real recovery.
Exactly. So our mission today is clear. First, we need to draw a really sharp distinction between that initial period of oxygen deprivation ischemia and its, let's say, less severe cousin, hypoxia.
A key distinction.
And then we'll dedicate significant time to detailing the 4 specific molecularly destructive mechanisms that kick in the moment blood rushes back. Understanding this double blow is just essential for anyone trying to grasp the challenges of modern critical care.
Okay, so to properly frame the severity of, say, a sudden blockage like in a heart attack, we need to start with some foundational definitions. In casual conversation, people often use hypoxia and ischemia almost interchangeably.
They do, and it's understandable, but in the context of cell survival, the difference between them is, well, it's profound, it's crucial.
So can you clarify that absolute distinction for us?
Sure. The distinction is absolutely crucial because it really dictates the rate and the severity of cellular collapse. Hypoxia fundamentally just means low oxygen supply.
Okay.
So the oxygen tension in the blood or the tissue is reduced. But crucially, in pure hypoxia, the blood flow, the perfusion, is often maintained. The pipes are still open, so to speak.
Right. So if the flow is maintained, even with low oxygen, the cell still has some kind of backup plan, doesn't it?
Yeah.
It does. If blood flow continues, the cell can still perform anaerobic glycolysis. Now, this process is incredibly inefficient. You only get 2 ATP molecules per glucose molecule compared to, around 30 or 32 from aerobic respiration.
What a huge difference.
Massive. But the key is that the necessary fuel, primarily glucose, is still being delivered by the flowing blood. And just as importantly, the toxic metabolic waste products like lactic acid are still being washed out and carried away.
Okay, so it keeps the lights dimly on.
Exactly. It allows the cell to sort of limp along, delaying irreversible injury, sometimes for a surprisingly long period.
But ischemia, that's a whole different ballgame.
Ischemia, yeah. The word itself means to hold back blood. It refers to a substantial reduction, or often a complete stop, of blood flow, usually due to a mechanical obstruction, like a blood clot in an artery, or maybe reduced venous drainage.
So the plumbing itself is blocked or severely compromised.
Right. And this is why it's significantly more rapid and severe than hypoxia. It compromises the entire delivery and removal system.
So if the plumbing stops, you're not just losing the oxygen needed for the main energy pathway.
Correct. Aerobic metabolism stops.
You're also simultaneously losing the supply chain for the backup pathway. The glucose delivery stops too.
That is the absolute core difference. Ischemia compromises the delivery of all the metabolic substrates needed for glycolysis. The fuel just stops arriving.
Yeah.
So not only does aerobic metabolism cease because there's no oxygen, but that backup anaerobic energy generation fails relatively quickly too because the glucose runs out or can't get there effectively.
And then there's the second fatal blow of ischemia you mentioned, the waste removal failure.
Precisely. This is critical. Toxic metabolites, especially lactic acid, from the cell's desperate attempts at anaerobic glycolysis, start to accumulate rapidly. There's no flowing blood to wash them out.
So the cell basically starts swimming in its own waste products.
In effect, yes. And this accumulation causes the intracellular environment to become highly acidic. The pH just plummets. And this acidosis, this drop in pH, It further inhibits the already struggling enzymes involved in glycolysis. It's a vicious cycle that accelerates the complete collapse of the energy system.
Okay, let's track that resulting chain reaction then. The cellular injury, starting with the immediate mechanical failure, the ATP depletion.
Right, so the initial event is universal, doesn't matter the tissue really. Oxygen tension falls. Oxidative phosphorylation, that's the main engine of ATP production in the mitochondria, it fails, and APP levels plummet.
Instantly.
Very, very quickly. And the first major functional consequence, this marks the beginning of what we call early reversible injury, is the failure of the sodium-potassium pump, the Na plus K plus ATPase pump.
Ah, the pump that maintains the cell's electrical balance. I remember that uses a ton of
A
ATP.
huge amount. It's constantly working, pushing sodium out and potassium in. So when ATP runs out, the pump just stops, dead in the water.
And the consequence of that stopping.
Well, the immediate result is an influx of sodium ions, Na+, into the cell. And because water follows salt, water H2O rushes in too. This is driven by the collapse osmotic gradient. At the same time, potassium K+ leaks out. So the cell swells up massively. You get cellular swelling of the internal organelles like the endoplasmic reticulum. You see changes on the surface too, loss of those little finger-like projections called microvilli, and the formation of membrane blebs, little bubbles pinching off. These are the classic morphological signs of reversible injury.
Okay, reversible for now. But if the blockage persists, the injury progresses beyond just swelling. You mentioned the pH plummeting. How does that acidic environment cause further damage inside?
That intense acidosis, combined with the ongoing energy failure, causes deeper structural changes. Things start to fall apart internally. Ribosomes, which make proteins, they detach from the endoplasmic reticulum, so protein synthesis halts. Inside the nucleus, the chromatin, the DNA complex, starts to clump together. These are all signs of severe cellular distress.
And if the ischemia continues even longer, then we hit irreversible damage.
Yes. If it's severe and prolonged enough, the cell crosses that threshold, the point of no return.
What specifically pushes it over the edge? What's the executioner, so to speak?
It seems to be a fundamental breakdown of membrane integrity, particularly mitochondrial membranes. We see massive mitochondrial swelling. The powerhouses are literally ballooning. And critically, there's a huge influx of calcium ions, K2 plus Fam-beta, both from outside the cell and from internal stores like the ER. The cell's normal mechanisms for controlling calcium are completely overwhelmed.
Calcium? Why is that influx so lethal, especially into the mitochondria?
Calcium acts like a central executioner. It activates multiple destructive pathways. It triggers enzymes that break down membranes, phospholipases, and proteins, proteases. Inside the mitochondria, this uncontrolled calcium, combined with some early ROS production from the struggling mitochondria themselves.
ROS, reactive oxygen species.
Yes, free radicals. That combination is often the trigger for the irreversible breakdown of membrane structures. It causes lysosomes, the cell's recycling centers containing digestive enzymes, to rupture. Those enzymes then start digesting the cell from within. That's autolysis.
Self-destruction.
Exactly. And ultimately, the plasma membrane itself, the outer boundary, breaches completely. Once that integrity is lost, the cell contents leak out. That's cell death via necrosis, a messy inflammatory death.
And it's not just necrosis, is it?
No. That mitochondrial damage also causes the release of a key protein called cytochrome C into the cytoplasm. And cytochrome C is a potent trigger for apoptosis, the programmed cell death pathway.
So the cell dies violently through necrosis and systematically through apoptosis. A double whammy even at the death stage.
Precisely. It's attacked from multiple angles, ensuring its demise. This is the landscape of the ischemic wound, the state of the tissue before we intervene. And this really exhaustive failure state brings us right back to the paradox. We must restore the flow. Reperfusion is clinically essential. It can save those cells that are only reversibly injured, the ones just swollen and blebbing. But the very act of reintroducing oxygen, nutrients, and crucially, circulating immune components, it simultaneously triggers this series of new damaging processes. And these processes cause the death of cells that might otherwise have recovered. This additional, potentially avoidable loss is ischemia-Reperfusion Injury, IRI.
Wow, it's such a critical lesson in pathology, isn't it? The environment we introduce to save the cell can, in that specific moment, be even more toxic than the deprivation itself.
It can seem that way, yes.
Okay, let's break down this double blow. You said there are 4 distinct synergistic mechanisms. Let's start with the immediate consequence of reintroducing that life-giving gas, oxygen. Mechanism A, oxidative stress.
Mechanism A, oxidative stress.
Right.
Right. When oxygen suddenly rushes back into tissue that is, remember, starved of ATP, highly acidic, has damaged mitochondria, and has depleted its natural antioxidant defenses, well, that oxygen is instantly transformed into a weapon.
How?
You get this massive acute spike in reactive oxygen species, ROS, and reactive nitrogen species, RNS. We commonly call these free radicals.
Where do they come from so quickly? How does the cell suddenly generate this T5 level?
There are several key sources kicking in almost simultaneously. First, those already damaged mitochondria. When suddenly exposed to oxygen again, they can't process it cleanly. They leak electrons during the reduction of oxygen, and these immediately form superoxide radicals, O2 soon. That's one major source.
Leaky power plants?
Exactly. Second, and this is really significant, during the ischemic period, an enzyme called xanthine dehydrogenase gets converted into a different form, xanthine oxidase, or XO.
Xanthine oxidase. What does it do?
Well, during ischemia, ATP breaks down into various byproducts, one of which is hypoxanthine. It just accumulates in the cell.
Okay.
When oxygen suddenly returns during reperfusion, this xanthine oxidase enzyme uses that oxygen to rapidly metabolize all the built-up hypoxanthine. And this reaction produces vast quantities of free radicals, specifically superoxide and hydrogen peroxide. It's like an oxidative bomb going off.
And you mentioned the cell's defenses are already down.
Precisely. The cell normally relies heavily on antioxidant systems, things like glutathione, superoxide dismutase, SOD, catalase. But these systems need energy, ATP, and certain cofactors to work, many of which were depleted during the ischemic crisis.
So a flood of free radicals hits a cell with no shield.
Exactly. The antioxidant capacity is overwhelmed. These free radicals then just run rampant. They attack and modify everything. Lipids and cell membranes get peroxidized, causing membranes to fall apart. Critical proteins get denatured, losing their function. Even DNA gets damaged. All of this just accelerates cell death.
Okay, that's a brutal start. Mechanism B, intracellular calcium overload. This builds directly on the ischemic injury, you said. Calcium started rising during ischemia, but reperfusion turns it into a lethal torrent. Yes. The calcium situation gets catastrophically worse during reperfusion. The cell is already injured on multiple fronts, remember?
Right, the membrane damage.
The cell membrane is already leaky from the ischemic swelling and maybe that initial free radical attack we just discussed. So calcium ions just pour in freely from the high concentrations outside the cell.
Okay, leaky outer walls.
And at the same time, those ROS generated in mechanism A directly damage the internal calcium stores the sarcoplasmic reticulum, or endoplasmic reticulum. These organelles normally hold calcium, but now they start leaking it into the cytoplasm too.
So the cell's internal reservoirs burst open, and the external dam has already broken, a double flood.
Exactly. You get this massive, uncontrolled rise in intracellular calcium. This isn't just generally toxic, although it is. It activates those destructive enzymes we mentioned earlier. But the really fatal step, the ultimate trigger for energy collapse, is what this high calcium does to the mitochondria.
Okay, what's the fatal step?
This excess calcium, along with the oxidative stress and low ATP, strongly, fatally favors the opening of a structure called the mitochondrial permeability transition pore, or MPTP.
The MPTP, let's pause on that because it sounds absolutely central to irreversible injury. What is this pore, and why is it opening the point of no return?
The MPTP is this complex channel made of multiple proteins that spans both the inner and outer mitochondrial membranes. Under normal conditions, it's closed, but its opening is triggered by exactly the conditions found during early reperfusion: high calcium, oxidative stress, and ATP depletion.
The perfect storm.
A perfect storm.
Yeah.
And when the MPTP opens, it causes two immediate, irreversible disasters for the mitochondrion and thus the cell.
Disaster one.
It makes the inner mitochondrial membrane suddenly permeable to small molecules, things it normally keeps out. This causes the electrochemical gradient across that inner membrane, the proton gradient, that actually drives ATP synthesis to collapse completely.
So it short circuits the power generator.
Instantly. It stops any chance of the mitochondrion making more ATP. Disaster 2 follows immediately. Because that gradient is lost, water rushes into the mitochondrial matrix. This leads to massive mitochondrial swelling. They balloon up and often rupture, spilling their contents into the cytoplasm,
Including that cytochrome C we mentioned earlier.
including cytochrome C, which then triggers the apoptosis. So the opening of the MPTP guarantees energy failure and initiates programmed cell death. It locks the cell onto an inescapable path towards death. It's a critical tipping point.
Devastating. Now we move to the body's wider response, which you said is often ill-timed and destructive. Mechanism C, inflammation. This is where the whole body gets involved, right?
Yes, this is where the immune system arrives on the scene, often making things worse. It's frequently referred to as sterile inflammation because there's no actual infection, no bacteria or virus.
So what triggers it?
The initial ischemic injury is messy. Dying and stressed cells release a variety of internal molecules that shouldn't normally be outside the cell. These act as alarm signals, known as damage-associated molecular patterns or DAMPs.
Danger signals.
Exactly. These like ATP itself floating outside the cell, bits of DNA, certain proteins. These DAMPs, along with some initial distress signals, cytokines, released by resident immune cells like macrophages already in the tissue, basically send out a system-wide alert.
Like a distress flare, calling in the immune system's cavalry.
That's a great analogy. That's precisely what happens. In response to these DAMPs and the low oxygen, the endothelial cells lining the blood vessels and the tissue cells themselves, parenchymal cells, they start expressing specific proteins on their surface called adhesion molecules. Think of them like Velcro patches.
Okay.
Names like P-selectin, ICAM1. When the blood flow resumes during reperfusion, it brings millions of circulating neutrophils. These are the body's first responder white blood cells, sort of the infantry of the immune system.
And they're designed to fight infection, which means that they're pretty destructive.
Highly destructive. They contain granules full of potent enzymes, and they generate lots of free radicals themselves. So these neutrophils encounter the Velcro, the adhesion molecules on the vessel walls in the injured area. They stick there, a process called margination.
They pile up.
They pile up, and then they actively crawl out of the blood vessels into the reperfused tissue itself. This is called extravasation.
So the immune system has correctly identified a site of damage, which is its job.
Right.
But its tools, the neutrophils, the free radicals they produce, the digestive enzymes they release, cause immense collateral damage in an area that's already incredibly fragile from the ischemia and the first two reperfusion hits.
Absolutely. This massive influx and subsequent activity of neutrophils causes significant additional tissue injury. They particularly damage the microvasculature, the tiny blood vessels, potentially leading to more blockages downstream, the no reflow phenomenon. And they damage the surrounding tissue cells directly.
Is there proof that this inflammation is really a major cause of the injury, not just a consequence?
Yes, we have compelling experimental evidence. In animal models of IRI, if you treat them with antibodies that specifically block these adhesion molecules, basically, you prevent the neutrophils from sticking and getting out into the tissue. You see a significant reduction in the overall tissue injury.
So stopping the inflammation helps.
It confirms that this neutrophil-driven inflammation is a critical, independent pathway driving ischemia-reperfusion injury. It's not just cleanup, it's part of the problem.
Okay, that brings us to the fourth mechanism. Mechanism D, activation of the complement system. You describe this as an amplifier.
Yes. The complement system acts like a powerful amplifier for all the damage we've just discussed. It's a cascade of proteins normally present in the blood plasma, part of our innate immune defense against pathogens.
How does it get involved here in sterile injury?
The setup is fascinating, and honestly still not fully understood. It seems that for reasons related to the altered cellular environment during the ischemic period, maybe exposed cellular components or altered proteins, specific antibodies from our own immune system, a type called IgM, deposit in the ischemic tissues during the period of no blood flow.
So they just sit there quietly while the tissue is ischemic?
Apparently so. They're inert during that phase.
Okay.
But the moment reperfusion occurs, the blood rushes back in carrying fresh complement proteins, particularly the initiating complex called C1.
And C1 finds the IgM.
C1 binds to these deposited IgM antibodies, and that binding is the trigger that activates the entire classical complement cascade pathway.
Okay, the cascade is triggered. What are the immediate destructive results of activating the system?
The cascade generates several critical, highly active protein fragments along the way. First, you get fragments like C3a and C5a. These are potent chemoattractants, they call, and even more neutrophils, drastically exacerbating mechanism C, the inflammation we just talked about.
So it fuels the fire.
Exactly. They also act as anaphylatoxins, increasing local vascular permeability, leading to more swelling and fluid leakage. But that's not all. The cascade continues.
What's the end result?
The terminal pathway of the complement cascade leads to the formation of something called the membrane attack complex, or MAC. It's formed from components C5b through C9.
The membrane attack complex sounds pretty literal.
It is. It's essentially a molecular drill. The MAC complex literally inserts itself into the cell membranes of the nearby injured tissue cells, forming pores or channels right through the membrane.
More holes in the membrane.
Yes. This further compromises membrane integrity, making the osmotic instability worse, exacerbating the calcium influx we talked about in mechanism B, and generally accelerating cell death.
So the complement system doesn't just add its own layer of damage. It actively ramps up the toxicity of the other mechanisms, the inflammation, the calcium overload.
Precisely. It acts as a powerful amplifier of the entire IRI process, linking the antibody deposition to widespread cell lysis and inflammation.
It's really a sobering picture. These four mechanisms, oxidative stress, calcium overload, inflammation, and complement activation, they sound like distinct molecular pathways on paper.
They are.
But in reality, upon reperfusion, they seem to converge almost instantly, creating this single, overwhelming wave of destruction.
That's exactly right. They feed into each other, creating a vicious cycle that can overwhelm cells that might have otherwise survived the initial ischemic insult.
Okay, so knowing that reperfusion is absolutely necessary, but it carries these four incredibly toxic risks.
Yeah.
The focus obviously shifts to mitigation. How do we disarm this biochemical time bomb that we ourselves trigger?
Right. And before we even talk about medical interventions, it's worth remembering the cell itself isn't entirely defenseless. It has evolved some protective responses.
Endogenous defenses.
Exactly. Mammalian cells can sense the threat of hypoxia and activate certain survival pathways. The best defined of these responses involves a master transcription factor called hypoxia-inducible factor 1, or HIF1.
HIF1, that sounds important. What happens when HIF1 gets activated by low oxygen?
Under low oxygen conditions, HIF1 protein becomes stable, it moves into the cell nucleus, and it switches on the transcription of literally hundreds of genes. These genes are all designed to help the cell adapt and survive the low oxygen environment.
What kind of genes?
Key among them are genes that promote the growth of new blood vessels angiogenesis, which could potentially bypass a blockage over time. It also ramps up the genes involved in glycolysis, maximizing that inefficient backup energy pathway.
Trying to squeeze out every last drop of ATP.
Exactly. And furthermore, it stimulates various anti-apoptotic or cell survival pathways. Essentially, HIF1 orchestrates this coordinated internal defense system to maximize the cell's tolerance to the ischemic stress.
Okay, given how critical HIF1 seems to be, I assume research is heavily invested in finding ways to boost it artificially, maybe to precondition tissues or enhance recovery.
Absolutely. Developing investigational compounds that promote HIF1 signaling or stabilize the HIF1 protein, even under normal oxygen, is a major frontier in research. The idea is, could we pharmacologically precondition tissue, maybe before planned surgery where ischemia is expected, or could we boost this pathway during an event to make the tissue more resilient?
Interesting. So that's the cell's own defense and research trying to leverage it. Now, turning to clinically useful established strategies, our source material highlights one non-pharmacologic intervention that seems remarkably robust, especially for the brain and spinal cord. That's transient therapeutic hypothermia.
Yes, inducing hypothermia, deliberately lowering the core body temperature, usually down to around 32 to 34 degrees Celsius, which is about 90 to 93. Fahrenheit is indeed the most widely accepted and proven strategy right now for mitigating ischemic brain and spinal cord injury, particularly after events like cardiac arrest or significant trauma.
Why does simply cooling the whole body work so well when the injury mechanisms seem so specific and molecular? What is the cooling actually doing at the cellular level?
It's effective precisely because it provides this multi-pronged systemic defense. It tackles aspects of all four IRI mechanisms simultaneously. Think of it as the ultimate global slowdown strategy.
Slow down.
By lowering the temperature, you fundamentally reduce the metabolic rate, the metabolic demands of all the stressed cells. This drastically slows down the consumption of any remaining ATP, slows down the production of waste products. Essentially, it buys crucial time.
Okay, so slowing metabolism helps generally, but how does it specifically counter those four IRI mechanisms we detailed?
Right. First, lowering temperature significantly suppresses the formation of free radicals. It slows down those enzymatic reactions like xanthine oxidase, hitting mechanism A.
Okay.
Second, it helps stabilize cell membranes, making them less leaky, which reduces the damaging calcium influx that helps with mechanism B.
Makes sense.
Third, it inhibits the release of inflammatory cytokines and reduces the expression and function of those adhesion molecules on blood vessels. So it dampens the whole host inflammatory response, hitting mechanism C.
Less neutrophil invasion.
Exactly. And by reducing overall inflammation and cellular stress, it likely indirectly dampens complement activation too, mechanism D. So all these factors together contribute to decrease overall cell and tissue injury simply by slowing down the kinetics, the speed, of that whole destructive cascade upon reperfusion.
It sounds incredibly powerful, just stabilizing the cell long enough for maybe its own repair mechanisms or the body's natural recovery to start kicking in.
That's the idea. Give the cells a fighting chance during that critical early reperfusion window.
But the source material was quite clear, and this seems to be a recurring theme. Reliable pharmacologic approaches drugs for reducing IRI consequences in routine clinical situations, like a common heart attack, remain a critical research gap. That suggests we need something maybe more practical or targeted than whole body cooling for every single patient, right?
That is the major challenge today, yes.
Oh.
Whole body cooling is effective, but it's logistically complex, has potential side effects, and needs to be initiated very quickly. And a huge issue with any IRI therapy is the therapeutic window. It's incredibly narrow.
Go narrow.
Often just minutes. The damaging cascade starts. The instant blood flow returns. So you need interventions that can be given right at the time of reperfusion, or even just before. This is why research has shifted so strongly towards these conditioning strategies. Preconditioning treating before ischemia is often impractical for sudden events like heart attacks or strokes.
Right, you don't know it's coming.
Exactly. So the real excitement is around post-conditioning, applying a protective intervention at the moment blood flow is restored, or in the very first minutes afterwards.
Okay, post-conditioning. Since we now know the four mechanisms, oxidative stress, calcium, inflammation, complement, what are the most current advancements trying to target these pharmacologically right at reperfusion?
Well, arguably the most exciting area, and where we've seen maybe the most promising translational data, focuses squarely on mechanism B protecting the mitochondria, specifically by blocking the opening of that MPTP.
Blocking the mitochondrial permeability transition pore, because if that opens, it's game over for the cell.
Essentially, yes, it's seen as a key point of no return. So the logic is, if we can pharmacologically block the MPTP from opening right when reperfusion starts, even just temporarily, we might prevent that catastrophic energy collapse and cytochrome C release, saving large numbers of cells.
And are there drugs that can actually do that? Block the MPTP?
Yes. Certain agents, most notably cyclosporine A and some of its non-immunosuppressive derivatives, have been heavily investigated. Cyclosporine A is already used clinically as an immunosuppressant after transplants.
Right.
But it was discovered, somewhat serendipitously, that it binds to a component of the MPTP complex, cyclophilin D, and inhibits its opening. Studies in animal models showed significant protection when given right at reperfusion, and has progressed into clinical trials for patients undergoing procedures like angioplasty for acute heart attacks.
So the principle is elegant. Keep the power station door shut during the initial storm of reperfusion.
Exactly. Keep the mitochondria functional during those first few critical minutes, and the cell has a much better chance of weathering the storm and recovering.
Okay, so targeting the mitochondria via the MPTT is key. What about addressing that explosive inflammation, mechanism C, blocking the neutrophil invasion?
That's another major therapeutic target, precisely because it involves circulating cells coming into the tissue.
Yeah.
It seems more druggable, perhaps, than processes deep inside the cell. Researchers are developing novel small molecule inhibitors and also monoclonal antibodies that specifically interfere with that neutrophil recruitment process.
How?
By targeting those adhesion molecules we talked about, the P-selectin or the ICAMs, preventing neutrophils from sticking to the vessel wall. Or by inhibiting the signaling pathways within the neutrophils themselves that tell them to activate and migrate. The goal is to allow the necessary blood flow of reperfusion, but effectively mute that excessive self-destructive immune response in the local tissue.
Makes sense. And what about the original source of much of the trouble, the free radicals from mechanism A? Are powerful antioxidant drugs still considered a viable part of a potential cocktail?
Antioxidant therapy. Well, it's proven notoriously difficult to translate successfully into clinical benefit for IRI.
Why? It seems so logical, just mop up the free radicals.
It does seem logical, but the challenges are immense. Finding an antioxidant drug that is powerful enough that can be delivered in high enough concentration to the specific site of injury at the right time and critically can effectively cross cell membranes and mitochondrial membranes to reach the actual sites of free radical generation. That's been a major hurdle. Many trials have unfortunately failed.
So maybe not a primary strategy anymore.
Direct, broad-spectrum antioxidant scavenging hasn't panned out as hoped. However, there's still interest in strategies that might boost the body's own endogenous antioxidant defenses, perhaps downstream of HIF-1 signaling, or drugs that target specific enzymatic sources of ROS, like xanthine oxidase or certain pathways in neutrophils. But it's proven tougher than initially thought.
Okay, so it really sounds like the overall trend, the way forward, is moving away from searching for that single magic bullet drug.
Definitely.
And more towards a combined, a multi-pronged strategy.
That is absolutely the consensus now in the field. Given the complexity, these 4 interconnected pathways all hitting the cell at once, future clinical success in mitigating IRI is almost certainly going to rely on a multimodal pharmacological cocktail.
A cocktail approach.
Yes, administering a combination of agents right at the time of reperfusion. You could imagine, perhaps, an MPTP inhibitor, like a cyclosporine derivative, combined with a potent anti-inflammatory agent targeting neutrophil recruitment, and maybe even an agent that supports the cell's own survival pathways via HIF-1.
Hitting multiple mechanisms simultaneously.
That's the goal, to intervene comprehensively during that very narrow critical window.
Yeah, and just briefly, does the duration of the initial ischemia affect how you deploy these strategies, or which tissues are most likely to benefit?
Oh, absolutely. The time tolerance for ischemia varies drastically between tissues. The brain is incredibly sensitive. Severe ischemia might only be tolerated for, say, 5 to 8 minutes before irreversible damage, widespread necrosis, sets in. This makes timely intervention and things like hypothermia absolutely critical for stroke or cardiac arrest.
And the heart?
The heart muscle is a bit more robust. It might tolerate severe ischemia for maybe 30 minutes, perhaps up to a few hours, depending on factors like collateral blood flow, before irreversible injury becomes widespread. This gives a slightly wider window for interventions like angioplasty and potential post-conditioning drugs.
And other organs.
Organs like the liver, kidneys, skeletal muscle, they can often tolerate even longer periods of ischemia, sometimes hours, before irreversible damage occurs. So this biological variability is key. The therapeutic strategy has to be timed and tailored to the specific tissue's known vulnerability and the likely duration of the ischemic insult. Conclusion. So if we synthesize all the insights from this deep dive, I think the most crucial piece of knowledge, the key takeaway for you, is that cell survival after a major ischemic event isn't a simple on-off switch. It's not just about restoring flow.
Yeah.
It's really about expertly managing the complex, dangerous molecular fallout that happens when that flow returns. Ischemia, the initial insult, causes damage through that dual collapse, starvation of fuel, and the toxic buildup of waste.
Right. And then reperfusion, it's the necessary poison. You have to do it, but it brings back oxygen, which immediately generates those destructive free radicals. It allows the blood back in, which exacerbates calcium overload, detonating the cell's powerhouses by opening the MPTP. And finally, the arrival of those circulating blood components triggers this massive inflammatory overreaction, amplified further by the complement system. It's a cascade.
It truly is. And it helps explain why our current best systemic approach, therapeutic hypothermia, is so effective, particularly for the brain. It works precisely because it recognizes this systemic complexity. It doesn't target just one thing. It broadly decelerates the rate of destruction across all 4 of those damaging fronts.
Yeah, the profound ability of just a simple transient drop in temperature to save potentially massive amounts of tissue, especially sensitive neurological tissue, it's really quite compelling.
It is.
But looking forward then, if we know that reperfusion injury is defined by this 4-pronged attack, oxidative stress, calcium overload via MPTP, inflammation via neutrophils, and complement activation, does it seem possible, maybe even likely, that future effective treatments, especially for very common events like heart attacks, won't involve a single drug,
I think that's highly probable.
but instead maybe a highly synchronized cocktail of therapies? Something administered at the exact moment of reperfusion, maybe even during the angioplasty procedure itself. A cocktail designed specifically to protect the mitochondria, maybe neutralize the most damaging free radicals or their sources, and decisively mute the immune system's damaging overreaction, all at the same time.
That synchronized, multimodal pharmacological tack that really represents both the ultimate challenge and, I think, the greatest promise of critical care research in this area today. Getting that timing, dosage, and combination right could move us much closer to truly saving all the cells that we fight so hard, often successfully, to reperfuse.
A fascinating, complex, and intensely critical area of medicine. Thank you so much for guiding us through this deep dive today.
It was my pleasure. Always important to understand these mechanisms.